Welcome, everyone. I am absolutely thrilled to guide you through this masterclass explainer on RNA therapeutics. We're directly adapting the brilliantly structured biochemistry lecture from Dr. Saima Barki to take you on a complete step-by-step journey through modern gene therapy and biological engineering. So whether you're prepping for an exam or you're just really fascinated by the future of medicine, you are in the exact right place.
Okay, let's dive right into this. Here's our roadmap for today. We'll start with the gene therapy landscape, then move to viral vectors, covering AAV and lentiviruses. Third is the RNA therapeutics revolution, followed by ASOs and self-amplifying RNA. Then we'll tackle the ultimate hurdle, delivery. And finally, we'll wrap up with the bioinformatics pipeline and a quick recap.
All right, starting right off with Section 1, the gene therapy landscape. So Dr. Barki actually starts her lecture with this really brilliant provocation. Think about it for a second. Suppose a patient has a mutation that causes a protein to be defective. How many different ways could we theoretically treat that disease? Our ultimate goal here is to achieve a true therapeutic effect, but here's the catch. We want to do this without necessarily changing the patient's permanent DNA. This is exactly how we're beginning to cure previously untreatable genetic diseases. You see, modern gene therapy isn't just about hacking the genome. We have an entire spectrum of intervention points. We could act at the DNA level to replace or edit a gene. We can act way downstream at the protein level to block or replace a defective protein. But right there in the middle is the RNA level. We can actually destroy harmful RNA, block its translation, or introduce brand new therapeutic RNA. This brings us to a really crucial conceptual distinction. On the left side, we have gene addition. Let's say you lack a functional gene. Well, we just add the blueprint to make a normal protein, which improves the phenotype. But on the right side, we have gene silencing. If your body is producing excessive amounts of a harmful protein, we don't add anything. Instead, we intercept the messenger, the disease-causing mRNA, using tools like siRNA or ASOs to silence it and reduce that harmful protein. It's basically like intercepting a rogue message before it can be read. Moving right along to Section 2, viral vectors, specifically AAV and lentiviruses. Now, what's really interesting about this slide is the sheer physical limits we're working with. Viruses are nature's perfect delivery vehicles, but they have strict cargo limits. Take adeno-associated virus, or AAV. It's highly favored in the clinic because of its low immunogenicity and high biosafety, but it has this tiny cargo limit of about 4.7 kilobases. Lentivirus, on the other hand, gives you way more room to work with, allowing for up to an 8 kilobase payload. Let's track the actual journey of an AAV vector into a cell. First, the engineered viral capsid binds to cell surface receptors. Step 2, it's taken up via endocytosis. Then it traffics through the inside of the cell. Step 4, the genetic material sneaks into the nucleus. And finally, gene expression occurs. But remember, these AAVs are designed as strictly non-replicating vehicles. The viral genes responsible for replication in disease have been intentionally stripped out and replaced with our therapeutic DNA. Okay, if you're taking notes, this is a must-memorize concept for your exam for sure. AAV delivers DNA that generally remains episomal. That means it just sits next to the host DNA without integrating, which makes it absolutely great for non-dividing cells, though cargo size and immune reactions are your main concerns. Lentivirus, conversely, stably integrates its genetic material right into the host genome. This is perfect for dividing cells because the daughter cells inherit the therapy. But it carries a major risk called insertional mutagenesis, meaning if it integrates in the wrong spot, it could disrupt cellular regulation and potentially cause cancer. Which brings us to Section 3, the RNA Therapeutics revolution. But what if we don't want to touch the DNA at all? RNA is just an incredibly programmable molecule. I mean, we've all heard of mRNA vaccines making global headlines, right? But they are literally just the tip of the iceberg. We have a massive toolkit at our disposal. We can use RNA to produce a protein, sure, but we can also use specific RNA structures to destroy a specific mRNA, alter splicing, or regulate massive networks of gene expression, all without ever altering the host genome. Let's zoom in on one of the most powerful tools we have, siRNA, or Small Interfering RNA. This is a highly specific, double-stranded molecule, usually about 21 to 23 nucleotides long. It enters the cell and loads into the RISC complex, specifically interacting with an Argonaute protein called AGO2. The complex cleverly degrades the passenger strand of the siRNA, holds onto the guide strand, and basically uses it like a homing beacon or a molecular hitman to find the complementary target mRNA, cleave it,
and stop that harmful protein from ever being made.
So how does siRNAs stack up against mRNA? Well, siRNA is typically designed therapeutically to be highly complementary to a single, specific transcript. mRNA, or microRNA, is endogenous, meaning it naturally occurs in your body, and it's about 22 nucleotides long. Crucially, miRNA recognizes its targets through partial complementarity, usually via its seed region. This means one single miRNA can regulate many different transcripts at the same time. That broad regulation is its natural strength. But therapeutically, it's a real challenge because of massive off-target effects.
Let's move along and see how this builds in Section 4, ASOs and Self-Amplifying RNA. While siRNA is double-stranded and uses that RISC complex we just talked about, an ASO, an Antisense Oligonucleotide, is a synthetic, single-stranded nucleic acid, usually just 16 to 20 nucleotides long. ASOs are fascinating because they can recruit an enzyme called RNase H to degrade RNA, or, amazingly, they can cause splice modulation. By binding to pre-mRNA, an ASO can actually force the cell's machinery to skip a defective exon or include a missing one. It fundamentally changes the protein produced without touching the underlying DNA.
Now, imagine for a second if your therapeutic RNA could actually photocopy itself. Well, that's self-amplifying RNA, or saRNA. Conventional mRNA is basically a 1-to-1 ratio. One mRNA makes a certain amount of protein and then degrades. But saRNA is engineered with an alpha-virus backbone encoding non-structural proteins 1 through 4. Once in the cytoplasm, these assemble into an RDRP complex, an RNA-dependent RNA polymerase. It grabs the conserved sequence element tags on the RNA and exponentially copies the RNA itself. This creates a prolonged, massive volume of therapeutic protein from just a tiny initial dose.
All right, Section 5, the ultimate hurdle, delivery.
Here's the harsh reality. Designing the RNA is actually kind of the easy part. None of this brilliant engineering matters if we can't get it into the cell intact. Just look at this timeline. Every single point here is a potential point of failure. You inject the RNA, it has to survive the bloodstream, it has to find the target tissue, it has to enter the cell, where it gets trapped in an endosome, and then it has to escape that endosome into the cytoplasm. This is incredibly hard, especially for central nervous system diseases, because getting these molecules across the tight blood-brain barrier is a massive pharmacological challenge.
Naked RNA just fails on its own. It literally gets chewed up by nucleases. Its negative charge naturally repels the cell membrane. It gets rapidly cleared by the kidneys. So what's the solution? Lipid nanoparticles, or LNPs. By encapsulating the fragile RNA in a specialized lipid bubble, we protect it from degradation, disguise its negative charge so it can enter the cell, and crucially, engineer the lipids to help it escape from that endosome so the RNA actually reaches the cytoplasm to do its job.
Finally, Section 6, Bioinformatics Pipeline and Recap. Let's see how this actually works in a real lab setting.
Before anyone even picks up a pipette, the work starts on a computer. Step 1 is pulling the exact transcript profile. Here, Dr. Barkey shows a search for the TP53 tumor protein on NCBI. You have to isolate the exact RefSeq mRNA sequence, in this case, NM000546.6. By identifying the exact conserved regions across different transcript variants, you make sure your therapeutic RNA targets exactly what it's supposed to, avoiding those massive off-target failures we talked about earlier. Once you've got those target sequences, you use design software to rank candidate siRNAs based on very strict thermodynamic principles. You need a GC content between 30% and 50% so the binding isn't too weak, but also isn't too rigidly stuck. You absolutely must avoid specific motif pitfalls, like strings of 4 Gs in a row. And you use structural predictors like RNA fold to ensure your target site on the mRNA is actually an accessible open loop, not buried away in a tight knot. Only the sequence that passes all these tests gets synthesized. And here we are, your masterclass finale cheat sheet. Seriously, lock this table in your memory. Conventional mRNA, that's transient. siRNA, transient and reversible gene silencing. ASOs, often prolonged, depending on the chemistry. AAV vectors, long-lasting episomal expression. Lentiviral vectors, stable, inherited genomic integration. And gene editing like CRISPR, that is a permanent genomic alteration. I want to leave you with one final provocative thought that tests exactly how deep your knowledge goes now. When designing an siRNA, why must the 5' end of the guide strand be A/U rich, meaning it has lower binding stability, compared to the 3' end? It's because this specific thermodynamic asymmetry is what literally forces the RIS complex to selectively load the correct guide strand rather than the passenger strand. It is just breathtaking molecular engineering. Thank you so much for joining me on this explainer, and keep exploring the incredible code of life.